I'm a Plankton taxonomist here at Cefas. So the purpose of this talk is we're going to cover 3 different groups of organisms. So I'm going to start with talking about phytoplankton, which is the plant component of the plankton. Then Nebula is going to move on to zooplankton and the work she does with that. And then Matthew and Peter are going to talk a little bit about benthic organisms. So I'll just share my screen. Okay, can everybody see that? Hello? What's going on?
Yeah, we can see it, Rob.
Okay, lovely. This isn't my presentation, or it was, but I didn't put this in there. Okay, never mind, it's a different one, so it's come up a bit strange. So for those who don't know, plankton are small organisms that float freely in the water column. I'm going to be talking about phytoplankton, as I say, which is the plant component of the plankton. And zooplankton, which Neville will talk about in a bit, is the animal component. So we analyse phytoplankton using high-powered inverted microscopes here at Cefas, using something called the Higgs-Mole method, which are these small plastic chambers. So we'll fill up our preserved water samples into these and slide this glass cover slip over the top, and then we'll wait for up to 12 hours for it to settle to the bottom, and then we view it through the base with the microscope objectives underneath. So why do we analyse phytoplankton cells? There's a number of good reasons. As a lot of you may know, they are fundamental to pretty much every marine ecosystem on Earth. You'll hear a lot of talks about the amount of oxygen they produce being comparable to rainforests and so on. And you know, if you're looking at fisheries, the phytoplankton provide food for the zooplankton, which provide food for the small fish, which provide food for the big fish, and so on. So they really are fundamental to pretty much anything that's going on with marine life. You use analysis of phytoplankton to provide an assessment of the water quality. So different species have different nutrient requirements. So if you have a lot of runoff from farms and fertilizers and things, that will affect the proportion of different species in the water. So if you're looking at things like eutrophication, they can give you an idea of that as well. Monitoring of potentially toxic phytoplankton is something we do quite a lot of here. So in any shellfish-based agriculture, and oysters that are filter-feeding organisms, they will take some of these potentially toxic species, and while the toxins themselves are not harmful to us when they're in the water, they can accumulate inside the flesh of these shellfish and build up to levels that can actually be quite dangerous for human consumption. So that's really quite an important area of work. And related to that, although not exactly the same, are harmful algal blooms. So when certain environmental conditions are met, some species of plankton will proliferate into huge blooms. You know, you're talking millions of cells per litre. And the fish can't respire properly, issues with oxygen depletion and things like that. So a lot of people are trying to look at things like predicting how these HABs, you know, when and how these HABs can occur. So that can be really quite interesting. They're also pretty. So we've got some examples here. These are some marine diatoms. Diatoms are a large group of phytoplankton made up with silica frustules, which is like a silica shell. So these here are examples of a species called Coscinodiscus, and this little sigmoidal thing here is a gyrosigma. So they're really quite pretty. We've got some odontella here as well. Again, these are diatoms. They sort of form these lovely little chains by linking up their little arms. And yeah, they're generally pretty cool. So, talking about looking for toxic phytoplankton, there are sort of 3 or 4 main groups we tend to look for in regards to monitoring shellfish harvesting. So a lot of these are what are called armored dinoflagellates. So I mentioned diatoms earlier. You have two overarching groups of phytoplankton mainly, which are dinoflagellates and diatoms. Now, some dinoflagellates are armored, and all this means is this clear area here is made up of cellulose, and it's like an armored plating around the outside of the cell. So when we talk about armored dinoflagellates, that's what we mean. This group here consists of mostly species called dinophysis, which are these pretty jug-shaped cells you can see here. And these are often linked to diuretic shellfish poisoning, which can get really quite serious. And you don't need too many of these cells for that to start becoming a problem. So as you can see, they're really quite nice. The key way we identify them is they've got these very characteristic body shapes. So for instance, Dinophysis tripos here has these lovely little legs that make it really easy to spot. And these structures at the top and at the side that look like little wings, these are called lists. And that makes it really quite easy for us to identify them. There is some variation in toxicity between these. So for instance, Dinophysis acuta here, I believe, is more toxic than Acuminata. Around the UK, though, we mainly tend to see Acuminata in coastal samples. Sometimes if you go more oceanic, you'll see things like this, which is always very exciting for us. Yeah, but that's the Dinophysiae. The next major group, and this is one some of you may or may not have heard of, is a genus called Alexandrium. These again are armored dinoflagellates. So you can see here on the right, they've got this sort of clear casing around them. That's that cellulose theca, the armor plating. These have been linked to producing paralytic shellfish toxins. So you may have heard of paralytic shellfish poisoning. It can very easily be fatal. So it's really important we look out for these. The key ways we identify these, they can be very tricky, but if you see here on the left, you've got this sort of indentation, this sort of Z-shaped indentation in the middle of the cell. This is called the girdle. And in Alexandrium, they're very deeply excavated. They sort of go right in and they've got big old lips on the outside. So that's often a good giveaway that something could potentially be an Alexandrium. Also around that girdle, you have this little line here, if everyone can see that. So this is a membrane going around the girdle, and that runs sort of through the middle. And yeah, there's a couple of key features like that we can use under microscopy to identify an Alexandrium. We can't really speciate them without further analysis. We can put them into species groups based on things like size. But in order to speciate, you need to look at this sort of armored plating, the cellulose plating I mentioned earlier. What we can do, and you'll see some pictures of this later, is called epifluorescence microscopy, which is where we use a stain called chalcofluor and then shine a mercury lamp onto the cells, and that causes the cellulose to glow blue, and it gives us a lot more definition of this armored plating. Is actually a diatom, so these silica-based cells I mentioned earlier. These are called pseudonychia, and again we put these into species groups. You would need electron microscopy to properly speciate them, but the key giveaways for these are they're long sort of lanceolate cells, and they have this really characteristic overlap here where the ends sort of join side to side. So these are three of the major groups of pseudonychia that we look for. So delicatissima are very, very thin and very, very straight, and they're quite easy to spot. Multistriata, they have this sort of characteristic sigmoidal shape to them, so they sort of flick off at either end. And then seriata are quite a bit larger usually and a bit wider, and you can see a lot more of the sort of cell contents as a result. So you've got these two chloroplasts either side of the center. This one is laying in a slightly different view, so you can't see that characteristic overlap, which is why you need a fair bit of training to recognize these easily. These have been linked to amnesic shellfish poisoning, which can get very serious and in some cases leads to memory loss and brain damage. but you really need a lot of these cells in the water for that to be a possibility. Now, I'm going to show you a video in a minute where there is a chain of this seriata, and there'll be some other thin chains of diatoms alongside, and you'll see that it can be quite tricky to know if you're looking at Pseudonychia or something else. So this little video is some live footage we've acquired from under the microscope. This species here is called a gymnodinium. This is another armoured dinoflagellate. This was sent to us from colleagues in Weymouth. And you can see there's a little flagella trailing out the back, which is one of the things these cells use to move. This cell here is called Porocentrum micans. Now, this isn't toxic, but they're just very pretty. They've got this characteristic sort of leaf shape. You can see it swimming around and me struggling to keep it in focus. And they have a very prominent spine sticking out at the front of the cell as well. But those are really quite common around the UK. We see them a lot of the time, and they're nice. The epifluorescence I was speaking about earlier. So you can see, I believe this was an Alexandrium, and you can see along the girdle that you've got much more definition of some of these cell features. So that makes it really quite a useful tool that we're trying to develop a bit more in the lab. Again, here you can see these plates much more clearly through epifluorescence. And again here, I'm going to have to skip the rest of this video, I'm afraid, because we will run over otherwise. But in general, epifluorescence is a really nice way for us to ID these armoured dinoflagellates. And not to mention, it looks really pretty. So speaking of the dinophysis I mentioned earlier, this is another group that can cause diuretic shellfish poisoning. These are called Porocentrum lima. You've got 3 cells here. And the way we identify those is their shape, and we also have these characteristic indentations at the top of the cell. It's quite difficult to see in this footage. The camera doesn't really do them justice. And they also have this central sort of pore. It's not a pore, but a central nodule here. Another thing you can use is, I don't know if you can make this out, but all around the edge of the cell are these little light patches, and the cell is covered in pores. And the pattern of these pores is specific to different species within the genus. So under fluorescence, or if you have an empty cell where the contents has been removed, then you can make out this pore pattern and identify the species correctly. So seasonality is really quite noticeable in a lot of these phytoplankton species. And what you can see here on this little animated GIF is the abundance of a species over the course of a year. So this is just a particular species of interest to us, and you can see it's really quite low up until early summer, and then it really spikes and we see a lot more cells showing up and then dies off again over the winter. And this can happen year on year, as we see here. So this is a 5-year time series of phytoplankton abundance. And you can see this is a bloom forming species. So these huge spikes we're seeing sort of later on towards 2018, 2019. This often happens when you get really good environmental conditions for that species to proliferate. Because these counts are so high, we are actually seeing cells in the winter months. It's just that because of the scale, the graph isn't able to show it. I've got one last thing to show you. So we monitor phytoplankton all around the UK. And this is a little animation I made a while ago. So I'll explain what it does. Any of the blue dots you see flashing up are where we've detected a species of interest below a certain level. And then as the animation moves through the course of the year, you'll see more and more different coloured dots flashing up, which represent species of interest above that certain level. And the larger the dot, the more cells there are. And you'll see it really does flare up over the summer. So this is around January, February now. It's sort of moving through the month. And then you can see as the year goes on, we start seeing more and more occurrences where things are really starting to warm up and we're getting a lot more of those cells, especially around Cornwall. This is partly due to sampling, although I would imagine there are also hydrographic and oceanographic features that phytoplankton are just better suited to down there. I know it's also a bit warmer, isn't it? So yeah, we have a lot of monitoring going on, and it's really quite good fun. So I'm going to hand over to Nevena now, and she's going to talk to you guys a bit about the work she does with zooplankton, and then we can have time for questions at the end. Okay. Thank you very much.
Right, she's Rob.
No
Just
worries.
get my thing.
Stop sharing. There we go. Over to you.
Right, let me just start it up. Okay, hopefully you can all see this okay. You give a shout otherwise if you can't. So I'm Nevena Almeida, a zooplankton taxonomist at Cefas, and I'll be covering the basically heterotrophic protist and metazoan component of the plankton community. So in other words, more of the animal side of things. So zooplankton, like phytoplankton, cover distances in the water column by drifting as opposed to swimming. As they feed mainly on the phytoplankton, they are also concentrated within the upper 200 meters of the water column, although most of them undergo daily patterns of vertical movement known as DL vertical migration. This is typically a single daily ascent, usually on either a nocturnal or twilight basis. This makes zooplankton especially significant in mediating fluxes within nutrient cycles, such as carbon. So there are several groups which zooplankton can be divided into. The crustacean zooplankton are the most dominant group, while some taxa are collectively termed as gelatinous plankton and include groups with extremely high levels of water and salt content in their bodies, such as jellyfish, ctenophores, and salps. Zooplankton are also further divided into either meroplankton or holoplankton, the latter of which remain in the water column for their entire life cycle, whereas meroplankton, by contrast, consist of the planktonic larvae of many aquatic organisms, which are otherwise endobenthic or epibenthic. And these can include also the larvae and egg of many fish species, which are collectively known as ichthyoplankton. So in this talk, I will just cover some examples of each of these groups, which we often encounter here at the Plankton Lab in Cefas, along with just some general key facts and features about each taxa. So first I will cover the crustacean zooplankton group known as copepods. So these are an example of a taxa which counts as holoplankton, as they spend their entire life cycle in the water column. The Calanoida order in particular is the most abundant and diverse pelagic copepod order, and these are the plankton which most frequently populate our samples, and the adults are generally larger than the adult individuals of the other main orders. So as a result, many commercial fish species are reliant on calanoid copepods within their diet, and calanoids themselves generally require phytoplankton as food, whilst other copepod orders may also be omnivorous or will also feed on bacteria. So I'll be playing a video next, and I apologise in advance if it starts off a bit jumpy. I know sometimes they take a little bit of time to get going smoothly. So hopefully if this is playing all right, this is video footage taken from the microscope. And most of the copepods displayed here are Calanoida copepods, particularly from the Calanus genus. So here you can see the trusty pokey stick being used to manipulate the specimens, as we normally would for identification purposes. So key features typically examined during this process would include such things as the number of segments certain sections of the body are divided into, and even the number of processes or setae on individual limbs. And calanoids have long antennules, as long as the body itself or even longer, and the adults are also generally easy to identify to genus level. And these morphological features can also be used to distinguish between male, female, and juvenile specimens. So here we have another type of crustacean holoplankton called euphorsids, which are commonly known as krill. These are a small group of shrimp-like plankton, of which some species occur in the greatest concentrations ever found for pelagic crustaceans. Therefore, many species are of considerable importance as food fish and some marine mammals. Euphorsids also notably possess 10 prominent eye-like photophores, which are unique among crustacean zooplankton and emit brilliant blue-green light. So hopefully you can just about see the photophores in the footage here, as the spots displayed along the length of their body. So the function of these photophores may relate to mating, social interaction or orientation, and as a form of counter-illumination and camouflage, which is used to compensate their shadow against overhead light. Euphorsids may additionally be distinguished by their anterior tinges of reddish pigment. They swim using their large legs, which are known as pleopods, and predate on other zooplankton by filtering them, as if with a feeding basket. Right, so to move on to an example of a meroplankton taxa, the plankton displayed here are from the family Nephropidae, the true or clawed lobster family. And this particular species is Nephrops norvegicus, which are commonly known as the Norwegian lobster and may even be enjoyed as scampi. In fact, they have even been known as the most important commercial crustacean in Europe. So being an example of meroplankton, only the larval stage is planktonic and lives within the water column. Therefore, the highest numbers of this species are found in the north of the European region, as its distribution is determined by the availability of suitable substrate for the adults to burrow into. So the larval stage of Nephrops, which is known as the zoea, is very distinctive with extremely long lateral spines. And as shown in this video, the pelagic larval stage is very distinct from the adult form, which has the more typical body shape of a lobster. The taxonomist wishing to identify the developmental stage of Nephrops will use the orientation of length of these spines to determine between different stages of zoea, which in the case of Nephrops is between 1 to 3. So moving on to ctenophores now, these are a type of gelatinous zooplankton and are differentiated from cnidarian jellyfish with their lack of nematocysts or stinging cells. They are commonly spherical or bell-shaped. You may have even often seen the spherical species stranded at the sea edge here in the UK, and these are commonly known as sea gooseberries. They are characterized by cells called cilia, which form rows like combs, giving them the term comb jellies. The rows of cilia beat like Mexican waves as such to propel the ctenophore along, also diffracting light as they do so. This gives off a beautiful rainbow-coloured iridescence, which you can somewhat see in the following clip, hopefully. So hopefully that's playing okay. Right, so finally we get on to the ichthyoplankton group, which is composed of the larvae and eggs of many teleos fish species. So these fish often produce huge amounts of eggs, up to hundreds of millions per female. The transparent pelagic eggs which are produced usually have a diameter between 0.7 to 1.5 millimeters, though in rare cases reaching up to 2.6 millimeters. So they're released into the water column after fertilization and may drift between the surface and the bottom during their incubation period. The planktonic larvae eventually hatch from the eggs and will go on to feed on phytoplankton and predate other zooplankton until they develop into the adult fish. Mortality of the larvae, however, is very high, potentially occurring from inadequate food sources, temperature, offshore transport and predation. As the survival and distribution of fish larvae and eggs has a big impact on fisheries recruitment, Cefas has been involved over the years in many surveys which aim to determine the abundance of ichthyoplankton around the UK. So as you can hopefully see in this video, some fish eggs can look pretty distinctive and beautiful, and these eggs shown here come from an offshore species of fish commonly known as pearlside, whose eggs also live up to their name with an iridescent and patterned shell. A lot of fish eggs can look very similar, however, and it's no easy task to be able to determine the exact species for every fish egg we come across. Sometimes this can be achieved, however, by measuring the egg diameter and oil globule size, if present, and paying attention to the pigmentation of the yolk and even of the developing fetus itself. And in this clip, I know some of these eggs were identified from fish species such as mackerel and dragonet. Similar principles are also applied when identifying fish larval specimens. As the fish larva grows, the pigmentation becomes more distinct. The larvae may also start to develop certain features once they reach a certain size, such as spines and developing fin rays. So here in this clip, larvae have been included from species such as mackerel, megrim and dragonets. So it does take time to develop the skill to discern these features reliably. Some fish species such as sprat, sardine and herring look identical and can literally only be distinguished by counting the muscle bands along the length of their body. But similarly, it is no small feat to be able to reliably identify other zooplankton groups in general. So having covered, sorry, I think I accidentally clicked out of that, but I was just finishing off by saying, having only covered just a small part of the zooplankton community, and just hope this helps to highlight the enormous diversity present within this community, as well as the importance for monitoring and the varied ways they can interact with aquatic ecosystems as a result. So on that note, I will be then passing over to the Benthic team, who will then be going through basically the benthic component of these aquatic ecosystems. So I'll just exit from this and hand over to Matt and Peter now.
Hello all. Hopefully you can at least see the presentation and hear me, I hope. I'm Matthew Curtis, a benthic ecologist here at Cefas. I'm just going to take you through a whistle-stop tour of what we do in the benthic lab up in here in microscopy. See, similar to phyto and zooplankton, we cover the macrofauna, which is anything over 1 millimetre that lives on the seabed. So it's a lot of the stuff you see obviously on the seashore. So yeah, we it can be anything from, you see, crabs, worms, everything. I think of species we get through the lab identifies probably just over 1,000 annelid worms, about 800 different crustaceans we get through the lab, 100 echinoderms and 600 or so different types of mollusks. So basically, I'll take you through this presentation. It's just a very quick coverage of how we process a sample. This is much better done in person, and hopefully one day, when all the restrictions are over, looking at who's in the list present, people are welcome to come and visit us, and we'll actually show you in person what we do. But for the moment, I'll just take you through it. So our samples are collected either on the Endeavour or other ships of opportunity, taken from grabs, and we get the pots of sediment back, and it can be very small pots, 0.25 litre bottles up to 20 litres of coarse sand, which is a nightmare to work through. So we have to then extract everything from these samples, and this is just just an example of what we do. So yeah, a little video showing a sample, very exciting shelly sand. Our first part is to extract take off the formaldehyde and float the sample to take off the lighter fauna. So we just put that over a 1 mil sieve. So we identify everything that's kept on a 1 mil sieve. And this is repeated several times to get rid of to take out as much of the fauna as possible. The next stage is the quarter fraction. We then pass over a couple of sieves to split it into into three fractions, a 5 mil, a 2 mil, and a 1 mil, just to make our processing a lot simpler. Because as you can see, trying to sort through that hash in one go would be a bit of a nightmare to pick out all the fauna, and is very inefficient. So we then, this sample split up into the 3 fractions. So these 3 sample fractions are then put into buckets and blasted again, floated again, so or any other fauna, loose fauna, is taken off, and then the rest of it is trayed up. And the floated fraction is then put onto Petri dishes and examined under a microscope. In case you didn't know what a microscope looks like, there you go. And this is just a bit of a video of down the microscope of the kind of bits we find on the Petri dish. So a worm, a Maldonid worm, Chinoderm. So that's all picked out and identification at a later stage, and then the rest is put onto trays, and we pick through this under illuminated magnifier. And everything then is brought into the microscopy lab for identification. That's where I'm sat now, and I will actually show you a bit of. I've got a camera looking live down the microscope at the moment, so I'll show you a few things shortly. It's just a little video that isn't playing of the microscopy lab. I'll skip past that. Yeah, so we've got plenty of literature to help us identify, and quite an extensive reference collection of all the taxa we're likely to see to help us go through this. And at the end, all this hard work, it just ends up in a nice species list matrix that can be used depending on what sites you collect it from. So we often do MCZ sites, dredge disposal sites, plenty of different areas. And now if I just shift over to my microscope, hopefully that's still showing. Okay, give me a second. Obviously, it's not on the right screen. So we stop sharing, share again. Hopefully now you're getting a live video view from down my microscope. So this is a sample I'm working on at the moment. It's just from Orford Inshore MCZ. So this is, unfortunately, the camera we use down the microscope does mean it's very zoomed in already. But this is just some of the worms that have come out of it. So these are it's a little Spirobranchus worm it was attached to some cobbles or some pebbles. And also in here is a couple of Savularia spinulosa worms, which are, most people might know, they are tube dwelling, but they can sort of have tubes on top of tubes and can form quite extensive biogenic reefs. And I think if Jackie's in the chat, she knows a lot about this after reporting a lot on the Norfolk sandbanks. So this is some of the worms in this sample. If I just move to another cell, which everything is very kindly moved out of the way. Move these back into the middle. Just a few other examples of when I can find them in the camera. For example, another polychaete worm. That's a little Schistomaringus. Sadly, I think we can manipulate it. It's got some. Should be able to. Well, if you were here, you'd be able to see some lovely jaws. You can actually see through his body. The camera's not showing that. And then a little Gamma-rid amphipod. Remember off the top of my head, I think it's Photis longicordata. So yeah, we have the fun of having to get down to all the sort of fine detail and to actually speciate it. I think my final thing in here is just a spatulid worm. And that's it from me. I'll pass you over to my colleague Peter Barry, who's going to talk about invasive species. So I'll just stop sharing.
Right.
Over to you, Pete.
Right, well, thank you very much, Matt. I'm using the same technology as Matt has. I've got a camera pointing down my microscope. And what you can see there on the screen is a bryozoan. This is, a bryozoan's not an unusual animal. We see a lot of them, usually stuck to rocks. What is unusual about this one is that he's stuck to a bit of litter that we found. You can see it's a scrap of plastic. The threat that's happening with floating plastic is that animals that should stay in one place or stay in their local environment have now been given rafts. They can move around on ocean currents. This is facilitating the spread of some potentially invasive species that they're okay, they're contained in their own environment, but then when they get out, they get into a new ecosystem. They end up in a new environment. Potentially they come to the UK and they can cause some damage. They can spread, they can outcompete native fauna. So this is something we really have to look into. We've just started investigating it at Cefas, and it seems like it's the right time. There was an article in The Guardian yesterday talking about potentially invasive species washing up here, attached to man-made objects. Some of the litter I looked through, it had bryozoans, it had mollusks, it had crustaceans, it had a lot of barnacles, a lot of sessile species, but also some mobile species, which don't need to stay stuck to the litter. Once they get here, they're free to move about and potentially settle. So I just wanted to draw your attention to that. And that's the end of our session. Thank you very much for listening. I'll hand you back to Rob.
Thanks, Pete. Right. Okay, so we've got some time for questions now. If there's anything people are particularly interested. Personally, I'd like to know a bit more about different things that can float in on plastics. I think that's pretty cool, but does anyone else have any thoughts? Sophie?
Yes, thank you. It was really good presentation. It really showed the richness of all these animals and plants and the taxonomy you have to go through. So how do you see taxonomy evolving in the future in terms of skills? We keep reading about how it's a discipline in crisis, so to speak, because we are losing the expertise, not just at Cefas, but everywhere in the world, really. So how do you see that moving forward?
Who's that directed at specifically? Is that a general question or anyone in particular? I know
It's a general question.
Yeah. Well, I know, Nevena, you've been working with sort of newer technologies with the zoo scan, haven't you? If you're interested in talking about that a bit, maybe.
Yeah, so yeah, there certainly has been a lot of talk about taxonomy being a discipline in crisis and taxonomists becoming endangered species, because I think basically there's been a recent drive towards like more large-scale multidisciplinary studies of oceanic ecosystems. So, and when you're looking into detailed like identification of plankton species, you generally like are very much having to focus on a smaller area because of the effort involved in identifying each of them in detail. So yeah, the question has been how to balance, you know, the need for taxonomy and plankton monitoring with, you know, being able to still basically be relevant for these large-scale multidisciplinary studies. So, you know, we've certainly seen image analysis techniques being developed recently, which, as you mentioned, Rob, ZooScan is a prominent one. There's FlowCam as well, and here at Cefas we've developed the Plankton Imager, which, you know, we continue to develop and iterate, and that one is particularly unique because that allows in situ monitoring on vessels rather than having to still take samples and process them through the machine. So basically, you know, you don't get as much high resolution taxonomy with those, but what they can do is allow, you know, plankton to still be included within these large scale sort of studies because, you know, these instruments are able to, you know, process samples like, you know, a bit quicker if you're willing to sacrifice the high resolution taxonomies. So, you know, it appeals to people because of the lower costs involved, and, you know, you're not necessarily even having to take physical samples with the plankton images. So, you know, I think the idea behind those is that, you know, it's not aiming to replace the microscope entirely, but to keep plankton in the spotlight still, so that, you know, basically people remember that plankton are still there, they're still important to be monitored, and that, you know, then we can also include microscope work alongside them. So I don't know if Matt and Peter would have anything to add on the Benthic side of things.
Certainly on our side, I mean, when I started at Cefas, there must have been 10 or more benthic ecologists, and up to a couple of years ago there was only one of us in the lab. Now there's a few more, so we're on the rise again. So it is always, because lab work's always expensive, it's always difficult to encourage people to do it in-house. So hopefully things seem to be on the improvement at the moment, and so hopefully taxonomy in Cefas is hopefully going to be on the rise. About the rest of the country, it's difficult to say. I know we have plenty of contractors that we work with and have done work for us, and they seem to be doing okay at the moment. So hopefully it's not too bad. Yes.
Thank you, Nevena. And Matthew.
Yeah, that's great. I would add one more thing though, in response to that, which is even with these sort of new emerging technologies that Nevena talked about, which are much faster but lower resolution, you still need people with the taxonomic skills and expertise to actually verify that it works. So there's always going to be a need for at least a few people, even if they're not working at the scopes, to actually understand all the little morphological structures. So I don't think it's going to totally die off anytime soon, hopefully. Anyone else? Any questions?
I mean, following up on that one, I was wondering, because we talk a lot about molecular technique, and effectively, if we were to use eDNA technique on every sample that we process, presumably we may not need the taxonomist. So do you see that happening, or
Well,
not?
I'm not an expert on eDNA by any means. I know a little bit about it from some blue carbon work I was doing. I mean, the main thing, as far as I see, is with eDNA, like, you don't necessarily know, as I understand it, I could be wrong, but you don't necessarily know that you're getting contaminants, especially in the marine environment, coming in from elsewhere, and you may just have a piece of a bit of zooplankton that's floated into where you're sampling. If there's a big feeding event, maybe somewhere further away, you could have some debris. Whereas if you couple that with taxonomy and you've got actual specimens in that area, then you know that the organisms you've sampled are actually supposed to be there. So I think you'll always need that verification. But I maybe don't understand eDNA well enough. I'm not familiar with the methods or the nitty-gritty things. But yeah, I don't know. We'll see.
Rob, from what I understand, is it's a very good tool if you're looking for a certain animal in an enclosed space. You can do a rapid assessment in something like a lake if you're looking for a certain fish. For a wider area, a big open area, it's a bit trickier. And I also think a lot of these molecular methods. Hang on, my camera. I can't get my camera to work. A lot of these molecular methods, they won't replace traditional taxonomy. They're complementary.
Yeah.
I don't think one is going to just replace the other.
I think that's fair. Actually, speaking of molecular methods, sorry, Sophie, we're doing some work in the minute in the Plankton lab where the genomic analysis has already been done, and then we're analysing the samples under the microscope. And we are finding quite a lot of differences between the two methods. So there's clearly a lot more work needed on both sides to marry that up, because neither one seems to be totally accurate yet.
Yeah, no, so I personally don't think that molecular technique will replace taxonomy. The reason I was asking is because just this morning I was reading this paper where it's for insect, but presumably can be applied to other things, where they combine capturing those insects with image recognition and eDNA at the same time. So which is why that got me thinking about it. But I see that as prohibitively expensive, potentially. And again, I think Jackie mentioned that we would need a library of every single potential species we may find to match them up.
Yeah, I think, sorry, coming in here, I think it would be, I think it's been proven quite useful for fish species, but I don't know how useful it has been for other smaller organisms.
Yeah, I see the question you left, Jackie, about whether we have DNA records of all species to enable eDNA, and I guess that's basically a similar vein to what Rob said, how you need the verification from taxonomy, I guess, to be able to collect those DNA records from the areas you're targeting. And obviously, as Peter says, new complications arise in open, large areas.
I think personally as well, part of the difficulty we're getting with taxonomy is it's rather niche and not really talked about in science education and things until you get to sort of the end of your sort of degree level education. So there's not really a lot of awareness, like for young people coming into their careers, which is a real shame because it's one of the few things where you actually get to look at the animals. So yeah, I think a lot of it is taxonomy maybe doesn't have the best PR. That's something I think would be a good direction to go if it's really a discipline in crisis, is to make it more appealing and more popular, because it's certainly effective, just not very well known. No worries, Jackie. Bye. She left already. Just seeing the chat.
Yeah, not sure if there are any other questions otherwise.
No, anybody else? I'm happy to sit and talk about plankton and things all day long, so it's fine by me. Okay, I think we'll leave it there. All right then, if that's everyone done, we'll make a move, and we'll see you all later. All 4 of you who are all in the next room. All right, thanks very much, guys, and yeah.
Thank you, Rob.
Cheerio, bye-bye.
Cheers, guys. Thanks a lot.